think we're ready for another episode of Endocrinology 2020, and we're continuing our theme on the regulation of metabolism. We covered all the gut hormones previously, and now we're going to start working on the big part of regulation of metabolism, which is the endocrine part of the pancreas. The endocrine pancreas is a rather large gland. Well, the endocrine part's pretty small, but the pancreas itself is a pretty large gland that arises embryologically from outpocketings of the embryonic foregut. And the gut is always endoderm in origin, so you get the idea that this is going to be endoderm. It begins as a series of ducts that end in secretory cells, and then other cells butt off of these ducts, and they form little islands in between the ducts that have no connection to the ducts themselves. So you end up with two different types of cells, duct cells and islet cells. In the adult human, the pancreas is located in the folds of the duodenum, extending from that curve of the duodenum all the way over to the spleen. So they often say it's in the arms of the duodenum. Very easy to find. The pancreas is a gland with both endocrine and exocrine functions. The large bulk of it is devoted to exocrine, and with a smaller part devoted to endocrine. Endocrine signals travel in the blood. That's why they're called endocrine. Exocrine products, which are usually inactive enzymes, travel in those ducts, and those ducts lead to the gut, so you can do digestion with those enzymes. The exocrine portion of the pancreas is represented by cells that are called acinar cells, and these secrete inactive digestive enzymes, which we call zymogens, and also secretes bicarbonate into the small intestine through the pancreatic duct. The bicarbonate is to neutralize the stomach acid that was produced earlier under gastrin and other control. The reason that digestive enzymes are secreted inactively is because if you put active proteases and stuff to travel through the ducts, they would eat the ductal cells. So you don't want those enzymes being activated until they actually get to the gut, where they're activated by pH changes in the gut. The endocrine portion of the pancreas is represented by small collections of cells scattered throughout the pancreas and consisting of only 1 to 2% of the total weight of the pancreas. So most of the pancreas is exocrine, only a little bit is endocrine. These collections of cells are called islets of Langerhans, named after the person who discovered them. In humans, there's 1 to 2 million islet cells, each one around 300 microns in diameter. Each islet has its own blood supply, so they're truly endocrine. Blood goes straight from the islet, drains to the hepatic portal system. The hepatic portal system goes straight to the liver, so blood goes straight to the liver from the pancreatic islet. And the major effects of the hormones from the pancreas are going to be on the liver and ultimately the rest of the body, but starting at the liver. The islet cells are innervated with both sympathetic and parasympathetic nerve fibers, so the autonomic nervous system has a big role in controlling the endocrine part of the pancreas. The anatomy of the pancreas, you can see there in the arms of the duodenum, you can see the gallbladder down there. The liver would be covering all this up. If you opened up an organism, you would see that the liver would be covering this up. The liver is producing bile that goes to the gallbladder and through the common bile duct that goes to the duodenum. The pancreas is in the duodenum, and as you see, the pancreatic duct dumps right into the duodenum. B shows you kind of how those the center cells are located relative to the ductal cells. And panel C there shows you just a close-up of the acinar cells, how they're arranged into these semicircles of zymogen secreting cells that lead into the ducts. They also produce bicarbonate that travels down through the ducts. And then the final panel D, shows how all of those acinar cells would be arranged with the islets of Langerhans being superimposed in between them. But there's just a lot more ductal cells than there are islet cells. The islets, on the other hand, are what we're interested in here. They're going to be the endocrine portion of the pancreas. Even though they only take up 1 to 2% of it, they regulate your metabolism to a huge extent. The various cells of the islet secrete a variety of signals that are going to enter the blood, and therefore they're truly hormones. The cells of the islets can be divided into types based on their staining properties and their morphology. There's at least 5 distinct types, of which you've probably heard of a couple. The alpha cells, beta cells, delta cells, F cells, and epsilon cells are the 5 types that are commonly recognized. The alpha cells average 30% of the islets and secrete glucagon. Beta cells average 60% of the islet and secrete insulin. The delta cells range between below 10% of the islets, and they secrete somatostatin. F cells are less than, they're 1% or so of the islets, and they secrete pancreatic polypeptide. And the epsilon cells are less than 1%, and they secrete ghrelin, which is a more recently discovered hormone. The composition of islets varies between species and at different ages and between regions of the pancreas. So not all islets are the same, not all of them have the same composition. For example, in the head region of the pancreas, there are a few islets that contain up to 80% pancreatic polypeptide cells. So they're not, there's a functional distribution of the types of cells and the hormones that they produce. A little bit more about pancreatic anatomy. You can see there's a blood supply right through the islets. The cells are in very close contact. So each islet has a blood supply running through it. There are beta cells, D cells, alpha cells, beta cells, D cells, PP cells, which F cells and epsilon cells in all these islets, but the composition varies. Different view of it, with all the exocrine cells out there on the outside with just the nuclei stained, and then the endocrine cells with the beta cells staining in red here, producing insulin, the alpha cells staining in green, producing glucagon, and the blue staining is the nuclei of any type of cell. Then there's pancreatic polypeptide cells, and somatostatin epsilon cells. And then there's ghrelin cells as well. This is kind of one way of looking at everywhere where we're going here, which is glucose metabolism, homeostasis. And this isn't exactly the way the world works, because we're not really actually trying to balance blood glucose all the time. Really what there is your body's kind of divided into 2 periods of nutritional availability. One we call the postprandial period. Some people call it the absorptive state. That's when you've just eaten a meal. And when you've just eaten a meal, you have a ton of blood glucose, so it's super high, and you need to put that away. It's just too high for normal functioning of the cells. You need to get it put into the liver, put into the muscles, put into the fat cells. You need to put that glucose away. And then the other period you have is a fasting period where you haven't eaten for a while, and you need to start freeing up some glucose because you're running out in the blood. So although they're drawing a balance here, and you're not really trying to keep it level. Whenever you eat a meal, you're trying to bring it down, and the rest of the time you're trying to bring it back up again. So notice that insulin by itself is what puts food away, nutrition away, particularly glucose here. And glucagon, adrenaline, growth hormone, cortisol, thyroid hormone, they all work to take it back out again later. So that's really, in a nutshell, everything there is about glucose metabolism and homeostasis. But the key hormone is going to be insulin. Insulin's the key to the absorptive state, the postprandial state. It promotes the uptake and utilization of glucose by almost all tissues. So insulin is hypoglycemic. Hypo means to lower. Glycemia means glyc means sugar. Anytime you see the word emia, it means in the blood. So it lowers the sugar in the blood. It's glycolytic, means it breaks down glucose. So by breaking down glucose, you can make fat available. It's glycogenic, which means it promotes the formation of glycogen. So you're going to break down glucose. You're going to store glucose as glycogen. And in doing so, you're going to lower blood sugar. because you're getting rid of the glucose. Insulin acts on muscle and other tissues, resulting in a net gain of protein. So you're going to take some of that glucose, you're going to take amino acids that are available around there, and you're going to build proteins. Insulin's going to act on the fat tissue, resulting in a net gain of fat. You're going to take circulating triglycerides, and you're going to use the energy from that glucose, and you're going to store it as fat. And then insulin effects can really be divided into two types that we need to talk about. Effects on membrane transport and effects on enzyme function. Membrane transport is how do we get the glucose into the cell. Glucose doesn't pass into the cell well at all without a transporter. So we need to have insulin create those transporters, get them into the membrane so glucose can enter. And then once it's in, you have to alter a bunch of enzymes so that you can store the glucose as glycogen, store the glucose as protein, store the glucose as fat. The effects on membrane transport occur in almost every tissue. Insulin increases glucose uptake in adipose tissue a lot, muscle tissue a lot. But interestingly, insulin doesn't change glucose transport into nervous tissue, and there's a reason for that. Nervous tissue has a glucose transporter built into it, and it's always enough of it there so the nervous system gets enough glucose. When you're totally running out of glucose in your body, the glucose transporters in the brain can still make sure that it gets enough, even when all the other tissues have been shut off. So your brain is specially protected to make sure that it always gets glucose. And your brain likes to use glucose as an energy source. Other tissues can use triglycerides and amino acids and work with those as energy. Your brain likes to use glucose as an energy source. Uptake of glucose requires assistance by membrane proteins. This is a type of facilitated diffusion. Insulin greatly increases the rate of diffusion into cells. It does not increase glucose entry into liver cells as directly as other tissues because liver cells already have a lot of glucose receptors. It increases it some, but not as much as other tissues. But muscle and adipose tissue, insulin greatly increases glucose. Okay, but insulin does create glucose to move into the hepatocytes, just not because of the transporters, but because it increases metabolism, which lowers the glucose inside the cell, increasing the concentration gradient, so glucose wants to go into the liver. In addition to glucose, insulin increases active transport of amino acids into cells, particularly muscle, so they can make protein, and it increases triglyceride entry into cell, especially adipose tissue, so you can make more fat. Enzyme metabolism effects, other than the transport effects, occur in most tissues. The greatest effects are liver, muscle, and adipose tissue, the three places where you're going to try and store glucose. The liver is going to store it as glycogen, muscle is going to store it as glycogen and protein, and adipose is going to store it as fat. The physiological actions of insulin in the liver. Insulin results in a decrease in key gluconeogenic enzymes so that glucose is not produced. The liver is one of only a couple of tissues that can make glucose from scratch, from straight out, and the liver, the kidney being one of the other ones. But you don't want to be making glucose when you have just eaten and you got a ton of glucose in your blood. So insulin shuts that down. Don't make any new glucose. No gluconeogenesis in the liver under insulin action. It increases the key enzymes to make glycogen. So you've got to get that glucose converted into, you have to phosphorylate it to make glycogen. And so there's one enzyme called glycogen synthase that's important in that. And then it inhibits glycogenolysis, which is the breakdown of glycogen. You don't want to break down glycogen while you're making glycogen, so it shuts that down. In the adipose tissue, insulin results in increased fat storage, increased triglyceride uptake, and in the muscle tissue, it increases amino acid uptake and therefore protein synthesis. It decreases ketogenesis. The liver makes a lot of ketones, and it shuts that down. Specific enzymes involved in the liver, the names of some of them, glycogen synthetase or synthase, it goes by two names, causes more glycogen to be formed. Glucokinase causes more glucose 6-phosphate to be converted into glucose 1-phosphate, and that can be used to put glucose away or to burn it. The increased breakdown of glucose and increased production of glycogen results in a low level of glucose in the liver cells. If you're taking the glucose and making it glycogen, or you're phosphorylating it, from the diffusion side of things, that creates a low level of glucose within the hepatocyte, and so that more glucose comes into the glucose transporters by passive diffusion. Excuse me for a second. In the muscle, insulin results in increased glycogen synthesis. Your muscle doesn't store as much glycogen as the liver does, but it stores quite a good bit, as any long-distance runner knows. Increased protein synthesis to make more protein. You shut down the enzymes that break down protein, and you quit releasing amino acids because you're trying to put away things, not release them. All resulting in the increased energy stores and increased protein in the muscle. In adipose tissue, insulin results in increased fatty acid synthesis. So you have fatty acids used to put fat away. Decreased fatty acid breakdown by inhibition of the thing that breaks down fat is called hormone-sensitive lipase. And insulin can shut that off. And increased lipoprotein lipase. This allows you to uptake fatty acids. You need lipoprotein lipase to free up the fats off of their transporters in the blood. So lipoprotein lipase takes the fats off of the transporters and allows them to come to the cell. And then increased glycerol phosphate synthesis, which is used in helping to put fat away. These all result in triglyceride or fat deposition. So what we're going to talk about overall, and what we've been talking about, is two loops controlled by two hormones. We have a low blood sugar loop. Low blood sugar causes the pancreas to produce glucagon. Glucagon is going to act on the liver and other tissues to free up glucose. Then we have a high blood sugar loop, which is your postprandial absorptive state loop, where high blood sugar promotes insulin release from the beta cells of the pancreas. Insulin goes to all tissues and to the liver to stimulate glycogen formation, ultimately lowering blood sugar. So this is the in a nutshell version of glucose regulation. There's a whole bunch of terms. I know it gets confusing, and they all sound the same because they all have gluc or glyc in them, but you got to sort them out. So I'm going to list them here, but I've been talking about some already, and I'm going to be talking about more later. So I'm not going to define each one here, but this is a nice page for you to go back to and write out some definitions. Glucose, glycolysis, gluconeogenesis, glycogenesis, glycogenolysis, glucagon, glycogen, glycemia. Those are all terms that you should be familiar with. And glycemia, usually there's a hypo or a hyper in front of it. Either too much blood glucose or lowers blood glucose, or too little blood glucose. Okay, glycolysis is a way of breaking down sugar. Remember that your body uses two ways of utilizing glucose, both of which start with glycolysis. Glycolysis cuts the 6-carbon sugar into 2 3-carbon molecules, which can either be converted into lactic acid, where you just stop after getting a couple ATPs out of the sugar, or you throw it into the Krebs cycle. And when you throw it in the Krebs cycle, you get 32 ATPs or so. But that requires oxygen. So we've got an oxidative pathway, which is the Krebs cycle, or oxidative phosphorylation, respiration, if you want to call it that. And then we've also got the shorter glycolysis, of which you only get a few ATPs, but it can be done in the absence of oxygen. So if you look at that, glucose is important. An enzyme called hexokinase is important for converting glucose into glucose 6-phosphate. So that's the step that's the phosphorylated form of glucose is not recognized in the diffusion equation, and the phosphorylated form also cannot cross through glucose transporters. It's got too much charge on it, it won't go through the transporters. So glucose entry is favored by converting glucose to glucose 6-phosphate. That allows a lot more glucose to come in because you're converting as soon as it gets there. Notice that you're actually burning an ATP in order to finally get some energy out of glucose, but that's for another place at another time. The important thing here is that glucose enters through transporters. Hexokinase transforms the glucose into glucose 6-phosphate. You'll see hexokinase called glucokinase fairly often. And then you can either transform it into lactic acid, where it can be stored for later, or it can be put through the Krebs cycle. If you put the pyruvate through the Krebs cycle, you get a lot more ATPs out of it. So high blood sugar increases insulin. Insulin causes the liver and the tissues to use and store energy, thereby lowering glucose. And then tissues both use and store, as we've already talked about. Glucose gets into the cells using transporters, glucose transporters, which are called glutes. There's another term for glutes, but we're not talking about those right now. They're a group of facilitated diffusion transporters. They work with the concentration gradient. They do not use energy to do this. And all have 12 transmembrane passes. We talked about, the serpentine receptors having 7. These things have 12 transmembrane passes. They have both of the amino and the carboxyl terminals end up inside the cell. So it's kind of an unusual situation there. But there's a whole series of them, and I'm only going to talk about the ones that are related to insulin, because there's a lot of them that in the kidney that are related to fructose uptake and things like that. So GLUT1 is associated with the bidirectional transport of glucose controlled by hormones such as thyroid hormone, but it's not a big insulin sensitive thing. So it's tissues that are going to take up glucose pretty much all the time will often have GLUT1, and then it's upregulated by other hormones besides insulin. GLUT2 is the one that's in the beta cells of the pancreas, and very important in insulin regulation. It's also in liver and kidney. On the hepatocyte membrane, so in the liver, it regulates entry and exit of glucose to and from the cell and controls hepatic glucose metabolism. But it's not really being regulated as much. It's upregulated by insulin, but not to the degree that some of the other ones are. In GLUT2 and intestinal cells and kidney tubule cells, there's absorption of glucose back into the body. So GLUT2, when it's in the intestine, is bringing glucose into the body. And GLUT2, when it's in the kidney, is reabsorbing glucose. You don't want to lose glucose. The only time glucose ever ends up in the urine is during diabetes, when you've overwhelmed the GLUT2 receptors, and they can't take it up fast enough. But you should never be peeing glucose. So you should be tasting your urine regularly to make sure that there's no sugar in it. GLUT3 is mainly present in the brain. It has a very high affinity for glucose. It's consistent with its function of transferring glucose into cells that need a lot of glucose and mainly use glucose. It's not under insulin control. So your nervous system always gets its glucose. It'll be, every other tissue will be shut down on glucose before the nervous system will be. Then GLUT4 is a strongly insulin responsive glucose transporter that's found in the heart, skeletal muscle, adipose tissue, and there are some in the brain. So the brain can upregulate glucose uptake, but it's got a lot of GLUT3 as well. But heart, skeletal muscle, and adipose tissue are the places that really get upregulated in the postprandial state when insulin is released. So how does this happen? Glucose goes through to GLUT2 receptors. Glucokinase converts it into glucose 6-phosphate, so that glucose inside the cell isn't seen, so more glucose continues to enter. Okay, so we're going to ultimately get insulin release from the beta cells, because insulin is in granules inside these beta cells. So this glucose 6-phosphate that's produced by the glucokinase enters glycolysis in the Krebs cycle, and it produces a ton of ATPs. For every molecule of glucose, gets somewhere around 32 ATPs. Those ATPs, as they build up and start to exceed the amount of ADP by some amount, 7-fold or so, actually inhibit a potassium channel that's located in the membrane. It's called ATP-sensitive potassium channel. It closes when there's a lot of ATP. Well, that causes membrane depolarization, and that causes the opening of voltage-dependent calcium channels. So those voltage-dependent calcium channels start letting calcium in. As you're probably aware by now, calcium is the leading signal for exocytosis of peptide granules, vesicles that contain peptides. So the insulin-containing granules that are lined up along the membrane fuse with the membrane that exocytose the insulin outside of the cell. So it's glucose itself, okay, that causes the insulin release. High glucose, a lot of insulin release. Low glucose, the ATP goes down, and this reverses itself, and you stop releasing insulin. So insulin release is directly driven by a glucose metabolic pathway, requiring the GLUT2 transporter. But as previously was thought that there was a little feedback upregulating the GLUT2 transporter, and that's somehow was leading to insulin release. But it's been pretty well shown that glucokinase is probably more important in regulating insulin release than is GLUT2. So, forgot about all those. Insulin is a peptide protein, protein hormone, peptide hormone. It has some internal disulfide bonds between what are end up to being two strands. It's also got another disulfide bond within one of the two strands. We call the molecule, after it's synthesized, after translation, it goes right into the endoplasmic reticulum, and we call that proinsulin. There's even a pre-proinsulin. So it starts as pre-proinsulin, proinsulin, and then ultimately insulin. So we end up with alpha and beta chains connected by disulfide bonds, and a C-peptide that is released in the process and doesn't appear to have any function. If you look at the primary sequence here, you'll see there are three disulfide bonds. The one in blue is going to be the alpha chain. The one in light gray is going to be the beta chain, and they're attached by a disulfide bond, and the alpha chain has an internal disulfide bond. And then the ones in darker gray are the connecting peptide, which apparently has no function. Pre-proinsulin is what you get right when you synthesize it, and that gets converted into insulin. And the C-peptide, this occurs partly, some of the cleavage occurs in the endoplasmic reticulum and the vesicles leading to the Golgi, but the final cleavages occur in the Golgi, and then it's packaged in the vesicles, leading to the exocytosis. Insulin action at the liver, where the glucose flows freely into the liver through transporter, where it's phosphorylated. Insulin binds to a tyrosine kinase receptor, a classic growth factor type receptor, except it's modified a little so you don't have to worry about dimerization. We'll see that in a minute. Then the intracellular effects of insulin are promote production of glycogen to store glucose, increase glucokinase expression in there at the level of the liver. So the liver is mainly going to be about glycogen storage and glucokinase, where you can burn more glucose. Glucose enters the liver via GLUT2, flows down its concentration gradient, then it gets phosphorylated by hexokinase, or if you want to call it glucokinase, and this helps maintain a favorable concentration gradient, so as long as there's glucose out there, it's going to keep flowing into the cell. And then the G6P can't go through the glutes and get back out. There's an ATP cost, though, because it takes an ATP to make glucose 6-phosphate, but you can gain that back through the Krebs cycle, or through glycolysis. This is a classic insulin receptor on the left, a similar IGF-1 insulin-like growth factor receptor on the right. We're not really interested in that right now. But notice that they're tyrosine kinase receptors, but they have an alpha and beta chain that are connected by disulfide bonds. And then they have a transmembrane region that's just an alpha helix that leads through the plasma membrane, and then an intracellular region that has tyrosine kinase domains. The difference between this and the classic like epidermal growth factor receptor, or one of those, is that it's already dimerized. There's already a dimer. It's already hooked together by disulfide bonds. But when insulin binds to it, it brings them closer together. It pulls them closer together. So it is kind of similar to the dimerization effect of growth hormone. And when it brings them closer together, those tyrosine kinase domains get activated and cross-phosphorylate. So you get autophosphorylation. So here's an example of what happens. Insulin binds to its receptor. It causes those tyrosine kinase domains to be activated, and then you get a whole bunch of tyrosine. There's I think eight or more tyrosines that get phosphorylated by the tyrosine kinase domains. Any phosphotyrosine can bind proteins that have SH2 domains, and then the SH2 domains will bind proteins that have SH3 domains, such as pi3 kinase, which is a big player here. One of those proteins that has, RS1 is a tyrosine phosphate binding protein, and it then binds to pi3 kinase and activates it. We'll get to that in a minute. Obviously, ATP is used enough to phosphorylate all those things. So this is really much a growth factor receptor story. It's the only difference is that it's already dimerized before insulin binds. There's two types of signals that you're going to get out of. Well, I guess there's probably three types of signals if you wanted to break it down that way. Glucose transport, where they have under metabolic signals, and growth signals. So insulin binding to its receptor is going to cause metabolic effects, and it's going to cause growth effects. And the metabolic signals go through these different proteins here that's called IRS. Anytime you see IRS, you know it's an insulin-related substrate for the insulin receptor. I think it stands for insulin receptor substrate family. One of the downstream effectors of that is pi3 kinase, phosphatidylinositol 3 kinase, things that happen when you activate pi3 kinase are glucose uptake through the glucose 4 transporter. So you're going to make more of the GLUT4, and you're going to put it into the membrane. Lipid metabolism, you start storing energy as fat, and you stop breaking down fat. pi3 kinase glycogen synthesis occurs, glucose to glycogen, and then protein synthesis in muscle. Lipid metabolism partly in the liver and partly in the muscle, in the adipose tissue. Growth signals go through this RAS, RAF, MEC pathway, causing cell proliferation and protein synthesis. So here's an example. How do we get the glucose transporter into the membrane? An unstimulated insulin receptor, and there's one little glucose transporter sitting there. You stimulate the insulin receptor, it turns through a pi3 kinase mediated signal transduction pathway, causes translocation of vesicles that have a whole bunch of glucose transporters in them. They bind to the membrane and leave those transporters in the membrane. So you get GLUT4 in the membrane as a result of insulin action. This is just another kind of overview slide like the previous one I had. I kind of like the other one better. But this is just showing you there's multiple effects of insulin binding to its receptor, GLUT4 synthesis and transfer to the membrane, enzymes and metabolic pathways, and gene expression, enzyme activities. Okay. And this one is more specific. I left this one in, even though it was redundant with the other two, because it shows you how many tyrosines there are. 1, 2, 3, 4, 5, 6, 7. So I see 7 tyrosines that can be phosphorylated there on that intracellular beta subunit. There's also an ATP binding site. There's a catalytic domain because you have to add the ATP to those tyrosines, break down ATP and add them to those tyrosines. So that's the functional part of it. Down below, you're seeing a couple of different pathways that are activated. One is the growth pathway on the left. Growth pathway has SH2 binding to GERB2 binding to SOS, then RAS, RAF, MEC, MAP kinase. And those are classic mitogenic, meaning causing cell proliferation, and protein synthetic pathway. So that's a growth pathway. Then on the other one, you'll see insulin receptor substrate pathway. That's probably IRS1, but there's a number of them. It gets phosphorylated, and in turn, it phosphorylates pi3 kinase. Then pi3 kinase turns around, and it phosphorylates phosphatidylinositol bisphosphate, our old friend from a previous pathway. But instead of PIP2 being degraded into IP3 and DAG, which you're used to, in this case, PIP3 gets phosphorylated. PIP2 gets phosphorylated into PIP3. So you have 4 phosphates on that one poor sugar, right? Phosphatidyl inositol trisphosphate. So there's 4 phosphate groups on one inositol sugar group. PIP3 then goes on and has effects, downstream effects, and then on the right you'll see that that stimulates that GLUT4 translocation pathway. There's something like 6 proteins involved in CAP to TCIO. TCIO is the final one causing the translocation of the GLUT4 transporter proteins into the membrane. So lots of stuff going on. PI3 kinase to glycogen. This is elaborated on that previous slide. Glycogen synthase makes glycogen in the presence of insulin. So how are we going to get that glucose to turn it into glycogen? If we have glycogen synthase when it's active and not phosphorylated, you can see that it will take glucose 6-phosphate and make it into glycogen. So that glucose 6-phosphate that's being made gets converted into glycogen. Excuse me. That's not the start of it. I'm sure it's not. All right. At least you can't get anything from me over. At least I haven't heard of anyone get anything over a video yet. So here we're making glycogen. Glycogen is long chains of long chains of glucose, which are made from glycogen synthase. Okay, so glycogen synthase makes glycogen, but it needs insulin to do that. In the absence of insulin, we have a different pathway. We have a thing called glycogen synthase kinase, which acts on glycogen synthase to inactivate it. So without insulin, everything to the right is not happening. Glycogen synthase kinase is converting all the glycogen synthase into phosphorylated glycogen synthase, and it can't do anything. So your liver doesn't make glycogen unless it has some insulin. Okay, so no PI3 kinase. This is mediated by PI3 kinase. So that's what the insulin's doing. It's turning on PI3 kinase. It's activating GSK. Then GS is inactivated by phosphorylation. Therefore, no glycogen. Now, the PI3 pathway activates GS. PI3 kinase ultimately inactivates GSK. So PI3 kinase, which was made in that signal transduction pathway we saw earlier, converts PIP2 to PIP3. Phosphatidylinositol bisphosphate into phosphatidylinositol trisphosphate in the membrane. These are membrane bound, and that then activates a molecule called AKT, and AKT is phosphorylated by PIP3. PIP3 then phosphorylates GSK. GSK goes on to phosphorylate glycogen synthase, and therefore you to inactivate. GSK has been inactivating glycogen synthase, but once you phosphorylate it, that doesn't happen. Sorry. So now you have active glycogen synthase before, in the presence of phosphorylated GSK. In the presence of GSK, you were keeping GS inactivated, but without GSK, GS is now active, and it can go on and stimulate that pathway of glucose to glycogen. Insulin action in the muscle. Insulin operates by binding to insulin receptors. As you see, as we saw, it's a tyrosine kinase receptor, and it activates a lot of different pathways. Intracellular effects of insulin promote glycogen to replace the glycogen that was lost during muscle use. So all day long, you're using your muscles, and you're converting glycogen to glucose to use those muscles. But you need insulin to make that glucose back into glycogen so your muscle has something to work with. So insulin is going to replenish muscle glycogen. Okay. So, and then it's also going to promote GLUT4 insertion in the plasma membrane to allow more glucose entry into the cell. So you have something to work with to make that glycogen. Insulin causes GLUT4, as we saw previously, but this is an elaboration of that. So we're going to have phosphorylated CAP that comes from the PIP3. And then there's at least 4 other enzymes that then turn on TCIO. TCIO takes GLUT4 containing vesicles, and those vesicles just fuse with the membrane. So you end up putting a whole bunch of GLUT4s, however many are in the vesicle, end up in the membrane. It's not done one at a time. That entire vesicle will get put into the membrane, just fuse with it. TCO is a type of what they call small T GTP binding protein, which means in its action it converts GTP to GDP. Well, we don't need to worry about that right now. And now you get glucose entry. Then glucokinase creates the phosphorylated glucose, which goes on to make glycogen. Now, this is sometimes a picture's worth 1,000 words. This is a cell before and after insulin. Before insulin, GLUT4 is being stained here in green. You see there's a little bit of GLUT4 in the membrane, but not very much. Most of it's being held internally in vesicles. But you hit it with insulin, and on the right, 30 minutes after insulin, all the GLUT4 is now in the membrane. So it's a huge translocation of GLUT4 in the membrane, allowing glucose uptake in these cells. So high blood sugar increases insulin, acts on the liver, fat, and muscle, converts sugar to fat and glycogen. That's half of our regulatory pathway. So I'm going to end with this slide, and then I'm going to begin another video that I'll post up starting with this slide. So this slide is called Opposition to Insulin. So besides insulin, there's a whole bunch of other hormones involved in energy metabolism. And all of those other ones oppose the actions of insulin and collectively called glucose counter-regulatory controls. So insulin is so powerful in its effects. When you eat, insulin just puts everything away and takes high blood glucose and turns it into low blood glucose. But when you're not eating, you need a variety of hormones to act under different circumstances to free that glucose back up so you can use it. So glucagon is the most significant. It's the most direct opposition. It's made by the pancreas, by the beta cells. But we'll see that the autonomic nervous system working through epinephrine, and then the adrenal cortex working through glucocorticoids, the thyroid gland working through thyroid hormones, and the anterior pituitary working through growth hormone are also important. Glucagon and protein synthesis is where we're going to start next time. So I'm going to end it right there, and I'll see you. I'll be posting this other video fairly soon. All right, bye.